Galveston Bay is alive and thriving. Hello, welcome to Seastar Base's virtual STEM to Stern program. My name is Carissa Laffey and I will be your guide today. I am an informal teacher inspiring people of all ages to preserve coastal and marine environments through art and science disciplines with a nonprofit in Galveston known as Artist Boat. Seastar Base Galveston has partnered with Artist Boat to provide a series of videos diving deep into the exploration of Galveston Bay aboard their vessel, the Bay Smart Express. Today, we are going to explore some of the unseen creatures that inhabit Galveston Bay. We will take a sample of the seafloor using a benthic grab and examine what marine organisms lay in the clay-like sediments. We're also going to try our hand at catching one of the most numerous and vital marine organisms, plankton. Finally, we will investigate turbidity in Galveston Bay. We are going to see how things that seem so small can have a mighty big impact. We will be deploying a benthic grab, which is also known as an Ekman grab, and it's going to sample the benthos environment of Galveston Bay. Benthos is the organisms that can be found living on the bottom substrate or sediment. The Ekman grab that I'm using is designed to sample areas that have a soft substrate. That means it doesn't have any rocks or sticks and leaf matter or dense vegetation. Now let's take a closer look at what the working parts are of the Ekman grab and understand how it works. On the Ekman grab, we have the jaws. These jaws open and clamp around our sediment sample. To prepare our grab for taking a sample, first we have to open up these jaws, and you have to open them one piece at a time and hook them to the top. As you can see, I'm being really careful where I put my hands, as these jaws are spring-loaded. Next, we are going to observe the hinge doors. The hinge doors allow for water to flow through the grab while I am lowering it to the bottom. Finally, the most important working piece of the Ekman grab is the messenger. The messenger is a weight that we will send down the line once the grab has reached that bottom substrate. The messenger activates those spring-loaded jaws and causes them to close at the same time, which would give us a good sample of that bottom substrate. When I pull the grab back to the surface, water pressure is keeping those jaws from reopening and losing our sample. In the sample, we can see two distinct layers of fine sediment. At the top of our sample, there's usually a thin layer of reddish-brown mud, while the remainder of the sample is just a dull gray color. Sediment in the Galveston Ship Channel is high in iron. And what happens when iron is exposed to oxygen for long periods of time? Well, it forms a compound known as iron oxide, which is also known as rust. That top layer of sediment is known as aerobic. Aerobic environments are characterized as having free oxygen. This plentiful oxygen binds with the iron to form iron oxide and causes the red or rust coloration of that top layer of sediment. But as we examine the dull gray-colored sediment, we know that it still has a high concentration of iron. But why is that soil not red? That's because the environment is anaerobic or lacking in free oxygen molecules. Now that we know a little bit more about that sediment, let's investigate one of the common native organisms found in that substrate, polychaetes. There are over 10,000 species of polychaetes, but the species that we are investigating today is sometimes known as the bloodworm. This name is attributed to the fact that bloodworms have a high concentration of iron in their blood, much like the surrounding sediment. This means that any small amounts of oxygen in the anaerobic soil easily binds with the iron in the polychaete's blood. We can see polychaete worms with our naked eye. To us, they appear as small red strings. But we will get a closer look with our compound microscope. To gather these polychaetes from the sediment, we need to use sieves. Sieves will allow particles of different sizes to pass through and leaves behind organisms or particles that are too large to pass through the sieve. You may have used a varied version of this while playing in the sandbox when you were young. For polychaetes, a 0.5 micron sieve will help isolate organisms of this size. Once we have identified one or more organisms, we'll talk about their morphology. Polychaetes, surprisingly, are one of the most common marine organisms. The name polychaete means many bristles. Notice the plentiful bristles, which are also known as parapodia, that line the body. Although their parapodia don't have joints like we do on our appendages, they still aid in movement like swimming, digging, or even walking along the seafloor. Polychaetes are segmented worms in the phylum Annelida, which also includes things like earthworms. Another notable part of the polychaete is the prostomium, or the head portion that bears the eyes, feeding palps, and tentacles. This is a region where there is an impressive amount of sensory reception. Most polychaetes in this region are detritivores, meaning that they feed on dead organic material in the sediment. But there's another organism that lives in the marine environment that you might not think about as much. Today, we will be deploying a tow to collect plankton. Plankton is any organism that floats with the waves, tides, and currents in the ocean and cannot swim independently of these forces. Plankton are small organisms and usually less than an inch in size. When they are by themselves, plankton are usually not visible to the naked eye. There are two main types of plankton, phytoplankton and zooplankton. Phytoplankton are microscopic plants that derive their energy through photosynthesis. These include things like diatoms, dinoflagellates, green algae, brown algae, and red algae. Zooplankton, however, are microscopic animals. Some zooplankton, known as meroplankton, are just the larval stages of larger marine organisms like fish, squid, crabs, barnacles, and shrimp, while others remain plankton for their entire life cycle, and these are known as copepods and isopods. Zooplankton living here mostly feed on phytoplankton. Galveston Bay's turbid and murky water has plentiful amounts of plankton due to its rich nutrient sediment. Our plankton toe is similar to the otter trawl net, but the mesh is much smaller, about 80 microns, and is more suited for collecting microscopic plankton. After we pull in our plankton toe, we will investigate which types have been found with our microscope. Make no mistake, plankton are the most valuable organisms in the marine environment. Because plankton provides the base of the food chain, any disturbance to its population, which could include change in the temperature of the water, the change in pH, or salinity, can cause a ripple effect of changes throughout all marine ecosystems. Take a deep breath in. Phytoplankton is responsible for producing about 70 to 80% of the world's oxygen, and it is produced in just the top 3 centimeters of the world's oceans. These organisms may be small, but they do have a mighty big global impact. What do you think when you approach a body of water that looks murky? Are you apprehensive about dipping a toe in? Today, we are going to be measuring the turbidity of Galveston Bay. Turbidity is a measurement of water clarity or the depth of penetration of light. The instrument that we are using to measure turbidity is called a Secchi disc, named after Angelo Secchi in 1865. It's interesting that in a world that has advanced so much since 1865 that we still use the Secchi disc today. To measure turbidity, we simply lower the Secchi disc into the water until we can no longer see it. It is at that point that we will take a reading. Notice that the Secchi disc has black and white coloration. The contrast between these two colors makes it easier to see when submerged in the water. The weight on the underside of the Secchi disc helps ensure that it sinks down instead of being carried off by the waves or currents. The markings on the line note meters so we can observe how far our Secchi disc is submerged in the water before it goes out of our sight. Now that we have measured the turbidity of this part of Galveston Bay, we need to know what factors contribute to turbid water and why it is important. What are some activities that you have observed around Galveston Bay that could contribute to a higher turbidity? There are several factors that include but are not limited to boat traffic, pollution, weather, sea state, and productivity or an abundance of phytoplankton and zooplankton. You may already hear lots of people refer to Galveston Bay water as brown and murky, and they're really not wrong. The issue is that we have negative connotations behind our words, and frankly, turbid water is not as bad as we make it out to be. There are several different reasons why Galveston Bay has a higher turbidity. One reason is that Galveston Bay is a soft bottom bay with clay-like sediment. There are 2 large rivers, the Trinity and the San Jacinto, that carry sediments into Galveston Bay. The other reason has to do with the sediment itself. Galveston Bay sediment is made up of really fine particles. These tiny particles don't need as much wave energy in the water to disturb them and suspend them in the water column. If you have ever visited places like Florida or the Caribbean and seen clear blue water, those locations have larger sand particles, so it takes much more energy in the water to disturb and suspend those particles, leading to much clearer looking water. But you may be wondering, what does turbidity have to do with plankton? Phytoplankton, in the form of algal blooms, can increase the turbidity of the water. An algal bloom decreases the amount of light that can reach the seafloor and uses the sunlight to fuel its rapid growth. Phytoplankton blooms are only a temporary phenomena because individual cells have a short lifespan, or the bloom will run out of nutrients that it was using to grow in the first place. Thank you for joining Sea Starbase on our virtual STEM to Stern program. Today, we explored the unseen in Galveston Bay by investigating a sediment sample for polychaetes and using a plankton tow to capture plankton. You got to witness how scientists measure turbidity and learned how turbidity affects the bay. We hope that you will join us again for another virtual tour of Galveston Bay.